Okay, so welcome everyone to the NPTEL course on Food Science and Technology. So this course is basically instructed by Professor H.N. Mishra sir, who is the professor in the Department of Agriculture and Food Engineering, IIT Kharagpur, and I am Shikha Sharma, a research scholar in the Department of Chemical Engineering, IIT Guwahati, and I am the TA of this course for the semester. Uh, so today is the live interaction session for week 6, and the assignment for week 6 is 4th of March. So please submit on time. So this week, so in this live interaction session, we'll be discussing about module 6, which is regarding micronutrients and bioactive compounds in food products. So its sources, structure, functions of vitamins and minerals, importance of micronutrients in the human nutrition and health, then stability of micronutrients during processing and storage, both phytochemicals and bioactive compounds, then pigments and colors, and then at the end, flavoring compounds. So this module 6 includes lecture 26 to 30, which includes 26 lecture includes vitamins, 27 is lecture is a discussion, and 28 is about phytochemicals and bioactive compounds. 29 lecture is about pigments and colors, and lecture 30 is about flavoring compounds. So I hope all of you have already gone through these 5 lecture videos discussed by Professor Mishra. So in this live interaction session, which we will be discussing these topics, and if you have any doubt, please feel free to ask. Okay, let's begin with today's session. So let's begin with the basic introduction of first topic, which is vitamins. So vitamins basically are organic compounds which are required in small amounts to perform specific biological functions for the normal maintenance of optimal growth and health of a organism. So vitamins are classified as fat soluble and water soluble. Fat soluble includes 4 types of vitamins: vitamin A, D, E, and K. And water soluble vitamins includes vitamin C, which is also known as ascorbic acid, and vitamin B complex, which is in and having 2 classes: energy producing and hematopoietic. So basically, in energy producing, we have vitamin B1, B2, B3, B5, B6, and B7, and folic acid, which is vitamin B9, and vitamin B12, which is cobalamin. They are in this class of hematopoietic. So now our first question for today's discussion will be regarding vitamins. So let's discuss that the primary role of vitamin A in vision is the formation. So we have 4 options here: rhodopsin, melanin, third is hemoglobin, and fourth is collagen. So anyone who would like to answer this question? I'm so sorry. So Fozia is saying A and Shrujna is also saying A, which is rhodopsin. So they both are correct. Rhodopsin is the correct answer here. I'm so sorry. There's some problem. Yeah. So rhodopsin is the first option. The answer is here rhodopsin. Rhodopsin is basically the basic vitamin A. When so vitamin A is basically used for we say that we have the vitamin E, yeah, vitamin A. So more of the vitamin A we should eat, which is good for our eyes. So basically, what happens in the eyes? What is the role of vitamin A? So its role is basically in the vision. It improves the vision and it forms basically this compound which is rhodopsin, which is in the eyes, so which is very important for human eye. So basically vitamin A, which is also known as retinol, it contains methyl substituted cyclohexyl ring. So this is the structure of vitamin. So this is this which is methyl substituted cyclohexyl ring. Also we can say it beta ionic ring, and there is this tetraene. This is the tetraene side chain which is having OH group, which is basically retinol. There is this OH group at the end, and for retinol there will be aldehyde group at the end. And if there will be retinoic acid, so what will be there? There will be carboxylic group at this particular position. This OH. In place of this OH, it can be any hydroxyl group, aldehyde group, carboxylic group, and if there will be ester group, so it will be retinyl ester at carbon 15. So this is the structure of vitamin A. Carotenoid as precursor for vitamin A. So carotenoid molecules, they basically act as a precursor for vitamin A. And this vitamin A is very important and it plays a crucial role in vision because it forms rhodopsin, which is a light sensitive pigment which is found in the rod cells of the retina. So this is the human eye. If we see in this, in the human eye, we know that we have this retina at the back side, right? This part is the retina. So here in the retina, if we see, so what happens? There will be different cells in the retina. So, like we know famous cells like rods and the cones are there in the retina. So, retina is formed from these rods, cones, right? So, they are basically rods and cones are important for detection of the light which is coming from outside. So, in our human eye, so the light will come through the eye lens and there are different other parts in the front of the eyes. So at the end, it will reach to the back part of the eye, which is basically known as retina. So in retina, we have the light sensitive basically cells, which are rods and cones. So what will be happening in this rods, particularly this rod cells, in this there are like this kind of structure will be there in that this rhodopsin will be present. So if we see in the pictorial way, so what happens is, so we were discussing that if we discuss this figure, so what happens, we have rods and cones as we discussed. So in rods will be disc-like shapes they will have. So this disc will have some visual pigments. So in this basically disc, there will be this structure of the cells, or the this structure is there, which is known as rhodopsin. So this is a rhodopsin molecule, which is present in the rods basically. So in a rod, each disc is an independent entity. So this, there will be in rods, there will be different disc-like structures will be there. So each disc will be basically an independent entity, and the outer segment of this disc, like this disc is having this outer segment, and it will have some elongated structures. So rhodopsin is basically kind of a cylindrical structure like this, and this rhodopsin is basically it enables basically the detection of low light conditions by undergoing conformational changes upon absorbing light, triggering a neural signal to the brain. So vitamin A basically maintains the integrity of mucus secreting cells in epithelial tissues, preventing keratinization and supporting the overall tissue health. They are also important in immune functions, which help fight against the infections by keeping this mucous membrane healthy. So, and also by acting as a barrier to the pathogen. So, not only so this rhodopsin basically, as we discussed, this rhodopsin help in like basically when there will be low light or the dark condition will be there, dim light will be there, so rhodopsin will going to and will go under some conformational changes, so that uh, and it will also send signals to the brain, so that brain will have also the knowledge that there is this dim light, and it can adjust our eyes. All these neurons will going to function, and all the other parts like iris, pupils, which is at the front, they will going to act if there is a dim light. So what happens? Our eyes, so the iris or the pupil of the eye, it will enlarge, and if there is more light, so it will contract. So that way, this whole function will going to happen, and this vitamin A will going to help with the formation of this rhodopsin molecule. Also, vitamin A helps the maintaining the integrity of mucus secreting cells. So, in our mouth, we have some on our tongue. If we see in our mouth, we will have mucus secreting cells will be there. So, in that basically, in that also, vitamin A helps in maintaining the integrity of these mucus secreting cells, which is there in epithelial tissues. But how? By preventing the keratinization and also supporting the overall health. So uh, vitamin A will going to help to uh, protect it from the this mucous membrane, to protect it from the pathogens or any infections. So it will going to help in that also. So here is the function, or here is a diagram which is telling how this rhodopsin molecule basically functions. So when there is like, this is the rhodopsin molecule. So when there is a bright light, so what will happen? This rhodopsin, the rhod visual pigments, bleaches in the light to opsin and all trans retinal. So basically, this rhodopsin will going to bleach into the opsin plus retinal when there will be high amount of the light. So all trans retinal basically is converted. Basically, this retinal is going to convert it into the vitamin A which is basically transported to the pigment epithelial and isomerized via this enzyme isomerase, basically 11 cis vitamin A. So what will happen? This cis retinal will first rhodopsin will going to be converted into the opsin and cis retinal when there will be light condition. So after that the cis retinal will transform into the trans retinal, and after that the basically it will be, this is the basically the reaction basically. So this trans retinal it is going to convert into vitamin A, which is basically transported to the epithelium, pigment epithelium. There will be isomerase will be there, which will convert it into cis retinal. So this basically cis retinal, this trans will convert into cis retinal, and finally again will be in the, will get the rhodopsin. So the 11 cis vitamin A is transported back to the rods and converted to 11 cis retinal, which spontaneously combines with opsin and to reform rhodopsin like this. So what is basically happening is rhodopsin is converting into all trans retinal, which is converted into all trans vitamin A. So with the help of isomerase, it will further convert into 11 cis vitamin A. This 11 cis vitamin A is converted into 11 cis retinol, and with the combining with opsin again. So it released the basically opsin before when it is in light condition, but when it is in dark condition, it will take again opsin with the 11 cis retinal, and it will form again pathway or the mechanism how this rhodopsin will going to help in the vision, or when there is bright light or when there is dim light, so how it is going to detect. So here is the role of vitamin A in the formation. This again the from this step to this vitamin A will going to, or rhodopsin will going to form. So vitamin A is going to help here. Then other options were melanin. So melanin is basically associated with the pigmentation of the skin or eyes. So it does not contribute basically to the vision process. So if we see here in this diagram, so we have melanocytes, which are the skin cells which produces the melanins. So these are the melanocytes. What they will do, they will produce melanin and transfer it to the skin, that will give the darker or the pigmented skin. And hemoglobin is basically involved in the oxygen transportation in the blood. So hemoglobin is basically a protein which is there in the red blood cells. So it is basically having 2 alpha chains and 2 beta chains, in which there is a heme group, and inside the heme group there is this iron. So next is the collagen. So collagen is basically a structural protein in the connective tissues, so it will not contribute to the vision. So this basically collagen is basically this kind of a protein which is formed from the amino acid. So it will form this collagen molecule and further the collagen fiber, and this collagen fiber will going to help in providing the structural or providing the, basically in the connective tissues it act as a structural protein. So in this question, rhodopsin is the correct answer. Rest other, which is melanin, is a basically pigment compound. Hemoglobin is carrying the oxygen, and collagen is a structural providing protein which is there for the in the connective tissues. So is this question clear to everyone? Can we move to this next question about vitamin D? So which of the following statement about vitamin D is correct? So first statement is about vitamin D3, which is all calciferol is synthesized from basically 7-dehydrocholesterol in the skin upon UV exposure. Statement 2 says that vitamin D2, which is calciferol is basically, and again the vitamin D3, they both, vitamin D2 and D3, require activation in the liver and kidneys to become biologically active. So which statement is our correct? We need to tell here. So here see that both statement 1 and 2 are correct. Anyone who would like to answer this question? Okay, let's move to the solution. So she's correct. Both 1 and 2 are correct. So vitamin D, basically it includes two vitamins, vitamin D3 and vitamin D2. So they are basically responsible for increasing the intestinal absorption of two minerals, which are calcium and phosphate, along with numerous other biological functions they'll perform. So they are also basically known as anti-rhechitic factors, the sunshine vitamin, which resembles basically steroid structure. So this is the structure of vitamin D. So vitamin D synthesis involves basically conversion of 7-dehydrocholesterol vitamin D3, which is cholecalciferol in the skin, basically upon the exposure of the UV light, specifically UVB. So UVB is from 280 to 315 nanometer rays, and there are two different other UVs. So there are basically three total UV. So UVA, UVB, and UVC. But here we are talking about UVB. So when there is the light of around 280 to 315 nanometers, so what will going to happen when this light will be there, fall on our skin? So basically conversion of this 7-dehydrocholesterol into vitamin D3. So statement 1, which is that vitamin D3 is synthesized from this molecule upon UV exposure in the skin, which is correct. Next, both basically vitamin D2. So vitamin D2 is derived from plants and vitamin D3 is synthesized in human body and can also be obtained from animal sources. So they are both inactive when we eat these vitamin from any plant source or any animal source, they'll go into our body and they'll be in inactive form. So that requires basically the activation, which will be activated by the sequential hydroxylation reaction for activation. So what will going to happen is vitamin, so we will first, like there will be two steps will be there in this process. One is the first hydroxylation, which will occur in the liver, which produce 25 hydroxy vitamin D, calcidol, calcidiol, and the second hydroxylation will also going to happen, which is there in the kidney. So there will be sequential two hydroxylation process. One is in the liver and another is in the liver. Oh, sorry, another is in the kidney. So this uh, uh, in the first hydroxylation, so we'll get 25 hydroxy vitamin D, and after the second hydroxylation, we'll get 1,25 dihydroxy vitamin D. Calcitriol will get. So basically, this is the biologically active form. So this activation process basically is essential for regulating calcium and phosphorus homeostasis. So if we see from plants, we get basically ergosterol, so which when get the light, it will form D2. And in animal, we'll get from animal we will get 7-dehydrocholesterol when we are exposed to the UV. So it will convert into D3. So it will convert it into active forms after the hydroxylation process. So if we see the pathway of vitamin D production, so what will happen basically because of the sunlight, specifically UVB, when it will fall on our skin, this 7-dehydrocholesterol basically it will convert into D3. When this D3, or if we have D2, so anything which is like from food we are taking D2, D3, anything, so it will convert into this 7-dehydrocholesterol in is converted to vitamin D3 via UVB. Vitamin D3 or D2 is further then hydroxylated in the liver to this molecule, which is 25 hydroxy vitamin D3, and further this from the second hydroxylation in the kidney, which will going to produce 1,25 dihydroxy vitamin D, the biologically active form. So second option is also correct. So both answer is correct here. Both the statement is correct here. So let's move to this next question, which is about the vitamin B12. So which of the following structural compound or the structural component of vitamin B12 is there? So is it the pyridine ring or corine ring with a central cobalt atom, or naphthocyanin ring, or isoalloxane ring? Anyone who would like to answer this question? The Fosya is saying B. Anyone else who would like to answer this question? That corine ring with a central cobalt atom. Okay, let's see answer. So she's correct that corine ring with a central cobalt atom. So vitamin B12 structure is this cyanocobalamin. Also we say this because this vitamin B12 is basically a unique among vitamins because it is having corine ring structure. Corine ring structure means which contains a central cobalt atom. So we can see here this is a cobalt atom in the center. So it is basically similar to tetrapyrrole ring found in the porphyrin compounds like heme in or heme with Fe and chlorophyll with Mg or magnesium, and basically it is similar to the structure of basically uh, like also we have this at the middle we have this magnesium ion. So here basically in vitamin B12 have this cobalt uh, atom is there. So and at the four sides we have the pyrrole units. This is the pyrrole units at the fourth side, and in the middle, this is a cobalt atom, which is basically forming the coordination bond here with the ligands. So the pyrrole ring have substitutes like, so this pyrrole ring can have substitutes like methyl acetamide and propionamide groups. And the basically this central carbon cobalt, sorry, I'm so sorry. The central cobalt atom basically is crucial for vitamin B12 biological activity. And this is basically bonded with 4 pyrrole nitrogen. So we can see this cobalt at the middle or at the center of the vitamin B12 structure. It is basically attached to this nitrogen pyrrole rings. So 4 pyrrole ring is there and 4 nitrogen is there. So it is basically attached to this 4 nitrogen from the pyrrole ring, which is forming the coordination bond basically with this a dimethyl benzimidazole DMB group below the plane. So here at the in the like below the plane there is this DMB group and a 6 substitute group above the plane. So there will be one this group DMB below the plane. 6 substitute group will be there above the plane. And this is basically vitamin B12 is basically is very important for DNA synthesis and enzymatic reactions. So the 6 substitute on the cobalt atom, it can vary. So it can be cyanide. So we can say it cyanocobalamin. So if there will be hydroxyl, we can say hydroxycobalamin. And if there is nitrite, we can say nitrocobalamin. And basically this vitamin B12 is synthesized only by microorganisms and not in the plants and animals. So there are sometimes we say that many people has this vitamin B12 deficiency because we cannot get it from animal or we cannot get from plant sources, and we will going to get it only from the microbial source. So in this question, option B, which is chlorine ring is there with a central cobalt atom. So let's see about other options. So pyridine basically ring, and another is naphtha quinone ring or isoalloxane ring. Basically, these rings are structurally components of different other vitamins like pyridine ring. So it is basically in vitamin B3 or niacin. So in vitamin B3, this basically is the pyridine ring, and naphthoquinone basically is there in vitamin K. So basically this particular structure or this particular part is naphthoquinone, and which is in vitamin K, and isoalcoxine is this one. So this is in riboflavin or vitamin B2, but it is not in vitamin B12, all these three. So only option is correct here. I hope this question is clear. Now we are discussing the next question. So in column 1, we have 4 different vitamins, vitamin A, C, D, B, and B1. In column 2, we have their deficiency disorders like scurvy, night blindness, rickets, and beriberi. So we need to match here. Anyone who would like to answer this question? So Fosia is saying B, which is this. Shiva D, Anjali D, Anju also D. Okay, let's see. So here answer D is correct, which is So these are the different structure of vitamin A, C, D, and vitamin B1. So let's see. So vitamin A basically it is basically crucial for the vision and epithelial health and also for immune function, and its deficiency may lead to night blindness. So we can see night before the eyes can be like this, but after night blindness it will look like this. And its deficiency also can cause to zero pathemia. Basically like this, eyes can go under, like there will be dryness in the eyes. So which is described by pathological dryness of the conjunctiva and cornea. So basically the eyes will going to be dried. So this conjunctiva, they will become dry, thick and wrinkled. So because basically in this night blindness will be there, vitamin E deficiency. Then second is xerophthalmia, which is the dryness of the conjunctiva. And third is the mucous membrane impact. So what will happen? The mucous membrane, which is present here in the esophagus. So what will going to happen? This when there is vitamin A in proper amount, what will happen? It will be healthy, this mucous membrane. But when there is vitamin A deficiency, the normal structure and the function of the cell will be impaired and it will become like. Then second is vitamin C, which is ascorbic acid, and it is a hexose or 6-carbon derivative, and it closely resembles monosaccharides in structure, and it basically consists of enolic hydroxyl group. So we can see here is the structure of the vitamin C. So this is basically having 6-carbon structure, or hexose is there. So it is a hexose derivative, and it has enolic hydroxyl group. This is basically the structure of this vitamin C. And the L-ascorbic acid basically is the active form of this vitamin. It acts as an antioxidant. It supports collagen production and tissue repair and enhances immune functions. It improves basically non-heme iron absorption, which is we get from plant sources, and aids in wound healings and uh, by contributing to the collagen synthesis. It is basically essential for immune support. So if there is deficiency of this vitamin C, we can have scurvy, where there can be fatigue or bleeding or swollen gums can be there, reddish spots on the skin, swollen muscles or joints, open wounds that will going to take a long time to heal, and falling of the teeth. So bleeding or poor wound healings will be their main symptoms if scurvy is there, which is vitamin G. And if vitamin D deficiency will be there, so it basically helps in calcium and phosphorus metabolism, and its deficiency can lead to rickets, which is in the children like this. So they can be deformed in the bones, and in the elder population there can be osteoporosis. So there will be reduced bone density, and because of that fracture condition can also happen. So we can see here the bone density is reduced compared to this normal condition. And vitamin B1, which is thiamine, it is basically a sulfur containing vitamin which forms TPP, which is thiamine pyrophosphate, basically pyrimidine ring and thiazole group, and attached basically through a methylene branch. So we can see the structure of this vitamin B1. So it basically forms, it can be found in like the forms like thymine, thymine pyrophosphate, thymine hydrothymine mononitrate, and main function is basically to take part in energy releasing reactions in the carbohydrates metabolism. So basically it requires, basically it is required for acetylcholine synthesis and also for ion translocation of the neural tissues, and basically this TPP plays important role in the transmission of the nerve impulses. So its deficiency disorder can be very severe and it results in beriberi. So it can be a neurological condition or cardiovascular issue related issues. So there can be the symptoms can include there will be redness in the hands or red spots in the hands or parts of the body, and which can be easily visible through the skin. So in this, first, which is vitamin A, is related to night blindness, which vitamin C is related to scurvy, vitamin D is related to rickets, and vitamin B1 is related to beriberi. Okay, let's move to this question. So the main forms of vitamin B2, riboflavin, NAD and NADP. Second is FMN and FED. C is coacetylase, CoA, and ACP, and D is TPP and PLP. So what are the different coenzymes which is uh, the form of vitamin B2 or riboflavin? So Shiva is saying C, Anjali is saying B. C and B. We have two answers here. And Anju is also saying B, and Jidnyasa is also saying B. So we have many B. So B is the correct answer here. FMN and FAD is the coenzymes basically here. So vitamin B2, which is also known as riboflavin, is basically a group of yellow fluorescent pigments called flavins. It is composed of 7,8 dimethyl 1 ribityl isoalloxane alloxazine ring structure. So this is basically this ring structure, which is we're talking about. So which is a heterocyclic structure with three interconnected, there are three interconnected rings, 1, 2, and 3, and they are not homogeneous, they are heterogeneous. And this isoalloxane ring is basically attached to D-ribitol moiety, which is this, through a basically nitrogen atom. So this from this nitrogen atom, this is connected to this D-ribitol. The coenzyme of vitamin B2 are basically FMN, which is flavin mononucleotide, which has phosphate group on the ribityl side chain, and FAD, which is flavin adenine dinucleotide, which is basically include an additional adenosyl monophosphate moiety. And riboflavin can exist in several oxidation states, including this yellow flavonoid, or second state can be flavosemic, red or blue depending on the pH, and also the colorless form, which is flavohydroquinone. Quinone. There are this FMN and FAD as the coenzyme here. And FMN is basically when we are, which is, what is this phosphate group on the ribityle side chain. So this will be FMN. Okay, and this FAD will be when there is this molecule which is basically adenosyl monophosphate moiety. So basically what is happening in the riboflavin, this riboflavin will go and it will be converted into its biologically active form, MN and FAD, through different enzymes like riboflavin kinase and then FAD synthase, like those enzymes. So first riboflavin will convert it into FMN, which is with the help of riboflavin kinase, and further with the FAD synthase, it will FAD. So these are basically the flavoproteins, which is important for different functions like DNA repair, chromatin remodeling, redox biology, protein folding, and bioenergetics. So basically these two coenzymes, FMN and FAD, they are crucial for redox reactions in metabolic pathways, including the electron transport chain. Also, it is important where it is going to facilitate the transfer of electrons. So this FAD and FMN, they are basically acting as a prosthetic groups in the flavoproteins, which are enabled for the redox reaction or oxidation reduction process, which are essential for the energy production. So like this, if we see riboflavin, it will go inside the cell or the mitochondria of the cell, and it will basically going to, with the help of FMN and FAD, this will be going to the riboflavin will go inside first. Basically, so riboflavin basically is imported basically here in the cells in with the help of some specific transporters, and then with that then in the cytoplasm basically this FMN and FAD will going to be formed, which is the basically the active form. So after that, what will happen? The FAD will going to basically this riboflavin basically is going to be transported into FMN and FAD, and it will take some of the ATPs here, and further it will go inside the mitochondria. So basically what is happening, riboflavin is from, suppose we are eating some vitamin B12, sorry, suppose we are eating some food which is rich in riboflavin, so that will first with the help of specific transporters will go into the mitochondria, and basically the first reaction will be in cytoplasm. So in cytoplasm, this FK molecule will going to convert this riboflavin into FMN, and it will take some of the ATP and it will convert into ADP. This further FMN is converted into FAD with the help of this FADS, and it will also take some of the ATP here. And in this mitochondria also some kind of this reaction will take place. And this FAD can also be transported into basically mitochondria or diffuse passively when the riboflavin concentration are high. So like this, it will take some of the FAD, sorry, ADP, and convert into ATP to ADP, and it will take some of the energy, which this energy will going to help basically in the processes, uh, like other like DNA repair and all other processes. So here, this FMN and FAD are basically the two coenzyme forms of vitamin B2. Okay, so which I hope so far is clear to everyone. So which of the following vitamins is least stable during heat processing? Vitamin D or vitamin B12 or A or C? Anyone who would like to answer this question? Shiva is vitamin C. Anjali D, vitamin C. Everyone? Anju also D. Okay, let's see the solution of this. So vitamin C is correct. So all of you are right. So it is basically the least stable during heat processing because it is highly sensitive to heat, light, and oxygen, and it is water soluble also. So when we are involving it into the cooking process, it will leach out basically, and structure is also very prone to oxidative degradation reactions when exposed to high temperature. So this makes to very challenging to retain this vitamin C during cooking, when we are doing any boiling or steaming. So vitamin C basically is prone to oxidation also when it is exposed to oxygen, and it can reduce the efficacy or potency of vitamin C when it is oxidized. So vitamin C is basically more stable in acidic conditions, and it will be degraded in alkaline conditions also. So vitamin C is basically the least stable during heat processing. Whereas if we talk about vitamin D or vitamin B12 or vitamin A, so vitamin A is basically sensitive to light, but all these are comparatively more stable under heat, because they three are fat soluble vitamins. Like vitamin A basically and D is fat soluble basically, and also their structure, if we see the structure of vitamin B12 or D or A, they are more robust chemical structure, and so which helps in the resisting of the breakdown of the structure of these vitamins when there is any oxygen exposure or heat exposure, or when we are doing any kind of food processing or cooking. So these are less prone to degradation. So if we see the degradation mechanism of vitamin C, so vitamin L ascorbic acid is the active form of the ascorbic acid. So it undergoes oxidations, or it can undergo other in when it like it go under oxidation, it forms DHAA, which is L dehydroascorbic acid, and further into the other products. And when it's in the human body, then what happens, it may have different temperature, light, oxygen, metal ions, pH change, water activity, enzymes, other different enzymes like ascorbic acid oxidase or peroxidase enzymes will be there. So because of that, all these enzymes, this L ascorbic acid will going to be lose its structure, and it may undergo several other forms or breakdown. So if we see here in this diagram, so above this line is the oxidative degradations, and below this is the non-oxidative degradations or non-oxidative pathways will be above the, and below are the oxidative pathways of ascorbic acid. So ascorbic acid can undergo into different structure changes, like in converted into DH or 2,3 diketo gluconic acids, or then basically erythrolose, or then further into 3-deoxy threosone. So there are like different molecules from ascorbic acids. It can undergo different like the transformation into different other molecules, and they can are further converted into more breakdown or more different compounds. And when we talk about the oxidative pathway, so what can happen, ascorbic acid first converted into DHA, so DHA may undergo and converted xylosones and different other compounds. uh, this 2,3 diketogluconic acid, one of the ascorbic acid, can convert it into threosone and further into different other compounds. So in overall, what I want to say here, that ascorbic acid is very like it's unstable, and it may convert into undergo oxidation, or it can undergo hydration or any other thing, or any other breakdown, like it can have the degradation due to temperature, pH, or light, or oxygen, or water activity, or metal ions, and it can be converted into other different other products. So it's very less stable. So here vitamin C, which is D option, is the correct answer. So let's discuss about our second topic, which is minerals. So minerals are basically the inorganic compounds. So they leave basically ash when burned, and they are converted into inorganic salts, organic compounds, and iron. So they are basically about 4 to 6% of the total body weight, and are present primarily in the bones. So in bones we have basically, so minerals basically supports bone and teeth structure, regulate enzyme metabolism, maintain acid-base balance and osmotic, and it helps in nerve and muscle function also in the human body. And there are the two different classification of minerals in our body, which are, first is the major minerals, or second is the trace minerals. So the first is the macro minerals, and second is the micro minerals. So macro or the major minerals are these: sodium, potassium, chloride, calcium, phosphorus, magnesium, and sulfur. And my minerals, which are the trace amount, so they are iron, copper, zinc, selenium, iodine, chromium, fluoride, magnesium, and molybdenum. So macronutrients, it is required around greater than 100 mg per day, whereas we need micronutrients than 20 mg per day. So based on the how much quantity we need to have in our body, it's classified into, if we need more, so we are classifying under macro, and if we need lesser amount, like less than 20 mg per day, it's considered under micro minerals. Okay, so let's discuss about the first question related to minerals. So column 1, we have 4 minerals: calcium, iron, zinc, and iodine. In column 2, we have deficiency diseases like goiter, anemia, osteoporosis, and impaired wound healing. So we need to match column 1 with column 2. Anyone who would like to answer? Shiva D, Anjali D, Kishore D. Anyone else who is answering this question, or can we move to the solution part? So Lakshmi C and Anju B. Okay, let's see. So we get B, C, and D as our answers. Okay, so B is the correct answer here. Let's understand why. So basically, calcium is basically in the 99% of this is stored in the brain as hydroxyapatite, and 1% calcium is there in the body fluids. And it is basically for the enzyme functions, blood clotting, nerve and muscle activities. And the calcium deficiency can lead to osteoporosis, where it is a condition which is categorized by the reduced bone density and increased risk of the fracture. And its deficiency can also lead to hypertension also. So we discussed this previously, that the bone density in the osteoporosis, it decreases. And iron deficiency, basically iron is its availability is around 3 to 5 grams total, which around 70% is found in the hemoglobin or myoglobin, and 30% is as the hemoside side why this okay hemosidrin. So basically most of our iron sources which we eat is there in the hemoglobin and the myoglobin. So basically iron basically helps in the transportation of the oxygen, and is important for cellular respiration and metabolism. So basically iron deficiency can lead to anemic condition, where what happens is there is insufficient amount of the hemoglobin production, and because of that there will be fatigueness and the weakness in the body. So this is a normal condition, and this is the anemic condition. So we can see there is insufficient hemoglobin production is there because iron is lesser. And we discussed that already, the structure of hemoglobin, that in RBC, which is disc-like shape, there is this hemoglobin. So here is the alpha chain, beta chain. So there will be 2 beta chains and 2 alpha chains. And inside this there will be heme group, and in heme group there is iron. So the symptoms of anemia may include that yellowing of the eyes. There is this chest pain and can have the spleen enlargement, and there can be the skin yellowing. So whenever you go to doctor and you have this anemic condition, they'll see your skin and eyes, and if your skin and eyes are yellow in color, or so they can understand that you have the anemic condition. And you can lose the muscles will be, you feel the weakness, or also you feel there is a change in the stool color, and you have the shortness of the breath, and you can feel more fatigue, and you'll feel like a fainting when you have this anemic condition. In the zinc, basically zinc is essential for the enzyme functions, nucleic acid synthesis, and metabolism. Zinc deficiency impairs basically wound healing, and because zinc is essential for enzymatic reaction in tissue repair and immune functions. So zinc deficiency basically can lead to loss of appetite, growth failure, skin changes, and reduced taste sensitivity. So basically this is the wound healing process is given. There are different stages of wound repair, and basically, so when wound repair begins with the hemostasis process. So first step is hemostasis, where what happens? Platelets, where a platelet plug prevents blood loss and a preliminary fibrin matrix is formed. So this fibrin clot will be formed where the blood is coming out, and there will be platelets will be there here, which is going to help to form uh, the temporary skin or temporary uh, uh, plug will be formed. Then second step is the inflammation, where to remove debris and prevent infection. So there will be this neutrophilic influx will be there, which is promoted by histamine release from the mast cells. And basically, monocytes arrives later and differentiate into tissues, macrophages to clear remaining cell debris, and neutrophils. So here there are different other uh, cells like mast cells and macrophages or neutrophils will come and they'll try to prevent the inflammation, and they will take the cells debris. Then next is the proliferation step. So where the carotenoids will come to the near the wound gap, and the blood moves through angiogenesis and fibroblasts and replace the initial fibrin clot granulation tissues. So first, what fibrin clot was produced as a initial solution to prevent the blood loss. Instead of that, there will be granulation tissues will be formed there. Then macrophages and regulatory T cells are also vital for this stage of healing. And finally, there will be remodeling. So in remodeling, what will happen? The deposited matrix is remodeled further by fibroblasts, blood vessels regress, and myofibroblast causes overall wound contraction. So at the end, remodeling will be there, where the overall skin will form inside. Next is the iodine. So iodine basically is essential for thyroid hormones. So thyroid hormones are thyroxine and triiodopamine. So its deficiency may lead to goiter. So goiter is this condition where the thyroid gland, we can see here, so there will be enlargement of this thyroid gland due to insufficient thyroid hormone production. So there is swelling in the throat or this neck area, throat area like this. So calcium is basically related here to C, which is osteoporosis. Calcium deficiency may cause osteoporosis. Iron may lead to anemic condition. Zinc deficiency may lead to impaired wound healing and is related to goiter. Okay, let's discuss this another question on minerals. So in column 1, we have 4 different minerals: magnesium, fluorine, copper, and iodine. And in column 2, we have their different roles. So in column 2, we have first option as protects teeth and prevents dental caries. The B option is component of thyroid hormone. C is maintains nerves and muscle functions, and D is essential for iron absorption and RBC formation. Anyone who would like to answer this question? Shiva, D, and Anjali and Kishore also D. Okay, many of us are saying D. So D is the correct answer. All of you are correct. So magnesium is basically involved in energy metabolism, enzyme activity, nerves, and muscle functions. And it plays a crucial role in nerves and muscle function by stabilizing ATP and supporting neuromuscular signaling. Exists as a free ion Mg+2 in the bodily fluids and tissues, and its sources may include plant foods basically, which is because it's basically this magnesium is in the chlorophyll structure. So magnesium basically will be at the middle, and it's coordination bond. Like we saw the cobalt is there in vitamin B12. So like that, it will form this magnesium will be at the central atom and in the chlorine structure. So magnesium is basically going to be help in the stabilizing the ATP and helping with the energy metabolism. So magnesium deficiency can cause the muscle cramping. And if we see the magnesium, so the glucose basically will be transported and it will further be converted into fat, glycogen and pyruvate. So this pyruvate basically uh, in this whole process, if we see when we are using glucose and we are sending inside the cell from the plasma to the cell membrane, if we are transporting this glucose, we need magnesium. And after this also, if we are converting it into pyruvate, we need magnesium. And then from in mitochondria, there are different processes like for production of the ATP synthesis process will take place where magnesium is required. So it is important here. And when we have the insulin as a molecule, so for get into insulin receptor, we need magnesium ion, magnesium as a mineral or atom we need. Also, like in muscle contraction, we need magnesium. So what happens? We in muscles we have this myosin. So what happened? Basically, this myosin is the inactive form. So it will convert into the active form, which is MLCK, which is basically myosin light chain kinase phosphatase or phosphatase will be added, and then further it will convert, like it will take calcium and it will convert to the active myosin, and it will add with the actin, and that will help to convert the into the ATP. So what will happen? Basically magnesium is basically going to help to convert the chemical energy which is the from the this pathways or this from this molecules, different myosin or actin, this molecules, uh, from these molecules, the chemical energy which is AT in the form of ATP is basically converted into mechanical energy. This chemical energy will convert it into mechanical energy, which is muscle contraction in this process. Fluorine is basically helps to protect the teeth and prevents dental caries by strengthening the enamel and resisting acid damage. And excessive intake, if we are taking of fluorine greater than 4 mg per liter water, it causes dental mottling in the children. So if we see here, this is the demineralization. So flake acid enters the rod in the teeth and breaks down the. So we can see this is the plaque acid which is going inside the teeth and breaking the enamel. But remineralization involves that fluorine will be there. So what it will do, fluorine enters the rod in the teeth to this enamel wounds or the lessons. So fluorine is help in the or the protection. Iodine is basically, we discussed in the previous question that it has a deficiency which is named as goiter, and it is crucial component of thyroid hormone, which is such as thyroxine T4 and T3, which is triiodothyroxine, and which regulates the metabolic processes. Copper is basically essential for iron absorption, aiding in hemoglobin formation and production. It is a cofactor for enzymes like cellulopsin. So basically, celluloplasmin. Basically, this is the molecule. Sorry, this is the enzyme. Basically, so what happens? Uh, Copper is attached to this, and it helps in the iron absorption. It enhances the iron absorption. So its deficiency may also lead to anemia. So if the copper deficiency is there, it may also lead to anemia. And skeletal defects, pigmentation can also be there. Reproductive failure also can be there, and cardiovascular diseases can also be there. So this molecule, which is this enzyme, so celluloplasmin, so its structure is that it is having 8 molecules of Cu plus. So greater than 90% of the copper to bind with this enzyme when we have the copper. So it is attached with this enzyme because one molecule is going to hold 8 copper atoms. So if we see copper metabolism, so if we are so we need basically 2.5 to 5 mg per day copper. So it will go into the so it can be, we can take copper from liver, from starfish, from nuts, from chocolates. We can get, and it can go into the stomach where around 25% is absorbed from in the small intestine, and it will go into the blood, and from blood it will be mixed with albumin, and further it will go in liver, and in the liver it will be stored like hepatocuprin. Basically, as we discussed, that the copper is basically essential for iron absorption. So basically, if we see the iron, how iron metabolized, so we can understand where copper role is this important. So iron, first it is absorbed from the dietary source. It is absorbed from in the small intestine basically, and it will go into the plasma basically, and basically from it will go to iron is regulated with the help of the liver. Basically, liver is going to regulate the iron, and when iron is utilized, so basically iron will going to be converted from Fe2 plus in from the this form Fe2 plus ceruloplasmin. So this molecule basically of enzyme is having 8 copper atoms. So here copper is going to help or to increase the iron absorption by because here from this enzyme it will going to help with the iron absorption in the whole metabolism of iron we are seeing. So here answer is D. So magnesium is basically here maintaining the nerves and muscle function. So basically with the help of stabilizing the ATP and supporting the neuromuscular signaling. And fluorine is related to protect the health and prevents dental caries, and copper is basically related to iron absorption and RBC formation, and iodine is related to thyroid hormones. So option D is correct here. Okay, let's discuss this next question now about sodium and potassium. So sodium and potassium work together to maintain what it maintains. Is it maintaining blood glucose level, or osmotic balance and nerve function, or DNA stability, or the protein synthesis? So if we talk about sodium and potassium, so sodium, which is around like 2%, potassium 5%, and fluorine 3%, they are basically key minerals in the body, and it is basically the sodium and the fluorine, they are the extracellular, while potassium cellular. So these all, so because sodium chloride is there, potassium chloride there is there, so their salts are there. That's why we are using chlorine, or also we are discussing here chlorine molecule. This sodium and potassium, they work basically together and they maintain. Shiva B, Anjali B, that osmotic balance and nerve function they are maintaining. So you both guys are correct. They are maintaining osmotic balance and nerve function. So sodium and potassium work for the balance of the fluid in and out of the cells, ensure proper nerve muscle is happening. So the potassium pump basically sodium out of the cell, and potassium will be into the cell, creating a gradient that helps send nervous signal and contract muscles. So because they are balancing this sodium and potassium amount, so that will going to help to maintain some gradient and further nerve signal and contraction of the muscles. So this balance is very important, and it keeps cell hydrated, and it prevents from swelling or drying of the cells. And sodium and potassium basically they are not controlling the blood sugar, and they are not stabilizing DNA and make the protein. So they basically support the process indirectly but not directly, because it keeps the cells healthy. So it supports these functions indirectly but not directly. Sodium and chlorine basically are particularly important for digestive fluids, while potassium is found in the lean tissues. If we see the sodium potassium pump, what it basically does is first Na plus, it binds to basically this pump, it will bind to the pump, and after that the pump basically is getting phosphorylated because of ATP will convert into ADP, and the pump will because of the phosphorylation it will change its formation, so that so the Na plus will going to release, and extra going to add to this pump. Basically these are the receptors. So this pump in from this pumps basically extracellular K plus will going to bind, will come into and it will basically help dephosphorylation. So when there will be dephosphorylation, the phosphate group will go out, and the pump will be coming to its original conformation, and the K plus will going to release from the pump inside the cell. like this pump functions. So here answer is B, osmotic balance and now function. Okay, so next question about minerals is which of the following minerals is most affected by the phytate content in the food? Calcium, magnesium, zinc or the iron? Anyone who would like to answer this question? C, Shiva, which is magnesium phytate content. Okay, let's see. So iron is the answer here. Phytates basically they are found in cereals, legumes, and some vegetables, and they strongly bind to minerals like iron, and they form the insoluble complexes with them, and that are difficult for the body to absorb, and significantly reduce the availability. While if we talk about the calcium, magnesium, and the zinc, they also bind to phytates, but their absorptions generally get less affected compared to iron because of the difference in their binding affinity. So iron will bind more strongly with the phytate compared to other minerals, and regulating mechanism in the gastrointestinal tract. So among the listed minerals, iron is basically more susceptible because of the non-heme iron, which is found in the plants and is more vulnerable to the inhibitory effects of phytates than the heme iron, which is found in the animal products and is less affected by such interactions. So in plant, the vegetarian people which iron they are eating from the plant sources, that is more affected by the phytates, and they'll form more strong bonding and become less bioavailable. So before consuming uh, iron, you should keep reminder that uh, in the food phytate content will be low as possible, otherwise it will inhibit iron absorption. So if we see the phytic acid, its structure is like that, this, and it is going to like this. It is going to trap this iron. Oh, sorry, trap this iron. So basically, this phytate is having this groups. This will going to bind this iron and become forms the insoluble and indigestible complexes. So if we see the structure and mechanism of action, so phytic acids, they are going to basically bind with metal ions, also with the proteins and also with the carbohydrates, and they will form carbohydrate phytic complexes or protein phytic acid complexes or metal ion phytic acid complexes. So they'll inhibit in the absorption of proteins and carbohydrates as well, and also with the minerals. So basically, these phytates, first phytate is the unavailable form, so it will convert into the more available form, which is inositol monophosphate, and that will going to basically that is basically found in many of the plants, and it will going to hamper the basically this basically immobilized phytate. It is basically going to bind with the free metal ion, and basically it will form inositol monophosphate and going to hamper the bindation or going to hamper the bioavailability of different uh, other molecules like carbohydrates, proteins, or metal ions. So iron is the answer here. Okay, so dietary fibers are known to improve iron bioavailability, gut health, and insulin sensitivity, then protein synthesis, calcium absorption. So basically, fibers are the phytochemicals, basically, which means that phytochemical means the bioactive compounds which are present in the plants, and they contribute to the color, flavor, and various disease-resistant related property in the plant. So, in the phytochemical class, there are different compounds like carotenoids, polyphenols, isoprenoids, phytosterols, saponins, and dietary fibers, and certain polysaccharides, and then glucose, enolates, and alkaloids. So here we are discussing about dietary fibers. So dietary fibers is responsible for which functions? Anyone who would like to answer this question? Lakshmi B, Anjali B, which is gut health and sensitivity. Okay, so you guys are correct. So dietary fibers are particularly soluble fibers like beta glucans and inulin. They are basically improve the gut health by promoting the growth of beneficial gut microbes, which produces short chain fatty acids that nourishes intestinal cells and reduces the inflammation. They also slow the glucose absorption, improve the insulin sensitivity, and aiding in blood sugar control. Their biological activity includes preventing the cancer, inflammation, hypertension, and cardiovascular diseases, and they improve the insulin sensitivity and gut health. They are found in carob beans, chicory, tamarind, barley, oats, and green beans. Not directly enhance iron bioavailability or protein synthesis or impact on calcium absorption minimal compared to other dietary factors like vitamin D or phytates. So it's basically going to help in the gut health and insulin sensitivity. So if we see that how dietary fiber helps the with the gut microbiota, gut health. So what happens is first, like if we have basically if we are eating dietary fibers, so first what is going to happen? Microbial enzymolysis of the fibers will going to happen. So microbial enzymes will going to act on the fibers, and then cross feeding of the gut bacteria will be there. Then the short chain fatty acids, they will be act as a substrate for the energy for colonocytes, and then there is absorption and circulation of this short chain fatty acids, and then in the host organs, and finally like this short chain fatty acids, they'll going to bind with the receptors and do the regulatory roles. So this is basically telling that the bacteria, basically these are the gut. So in this gut we have the bacteria which is going to basically first with the help of the enzymes it will break down the fibers, and further it will convert it into short chain fatty acids, which is going to basically help with the regulation of the receptors and other things. So basically answer B is correct here. Gut health and insulin sensitivity improves by the dietary fibers. So next is about the flavonoids. So basically which of the following statement about isoflavon flavones is our correct. So statement, we have two statements, statement 1 and statement 2. So statement 1 says isoflavones are a subclass of flavonoids, and statement 2 is saying they mimic estrogen in the body and may reduce the risk of hormone related cancer. So which statement is correct here? So basically, this isoflavones, they come under polyphenols. So basically, polyphenols are the natural compounds which are having this phenolic structure. So two types, flavonoids and non-flavonoids. This isoflavones, basically, they are in the flavonoid category. And in non-flavonoid, we have stilbenes, tannins, other compounds, and the anthocyanins and isoflavones, they will be in flavonoids. So which statement about isoflavones are correct? Everyone is saying C. Okay, let's discuss. Both 1 and 2 are correct. Correct. So isoflavones, basically they are two compounds, genistein and diazine. So they are the basically subclass of flavonoids, and they are the group of polyphenolic compounds which are found in soya or legumes. So isoflavones, basically they exhibit phytoestrogen activity, which means they mimic estrogen in the body by binding to the estrogen receptors. So this activity basically can modulate hormonal balance and potentially reduce the risk of hormone related cancers like breast cancers or prostate cancers by either enhancing or blocking estrogen effects depending on the hormonal environment. It also possesses antioxidant, anti-cancerous, antimicrobial and anti activities. So answer C is correct here, that both of the statements are correct here. So next, the following phytochemicals is classified as a still bean: reversatol, lycopene, catechol, catechin or limonene. So basically, still beans is non-flavonoid compounds as per this classification in the polyphenols. Anyone who is answering this question? Anjali A, resveratrol, Shiva B, lycopene. Okay, so Anjali is correct here. So resveratrol is the correct answer. So basically, resveratrol is classified as a still bean, a type of phytochemical characterized by 1,2-diphenyl ethylene structure. So this is having two phenyl rings like this. Two phenyl rings are there connected with a vinyl group. Sorry, vinyl. So this is vinyl and this is double bond. So vinylene group is there, and at the end two phenyl groups or phenyl rings are there, and it is primarily found in grapes, red wines and berries, and is known for its antioxidant, anti-inflammatory and cardioprotective properties. Still beans, so it is basically like resveratrol, can modulate gene expression against oxidative stress. And if we see the lycopene, which is a carotenoids, it is responsible for the red pigmentation in tomato associated with the prostate health. Next is the catechin. So catechin is found basically in the tea or the cocoa. So is a flavonoids, and which is having antioxidant properties. And limonene is basically found in the citrus peels, and it is having digestive health benefits, effects, and antimicrobial benefits, and is basically a terpenoids, which is found in citrus peels. So here resveratrol is the correct answer. The tannins reduce the bioavailability of minerals by first binding to lipids and soluble complexes, second chelating metals, ions like iron and zinc, third blocking protein synthesis, fourth enhancing oxidative stress in the gastrointestinal tract. Shiva B, Anju B. Chelating metal ions like iron and zinc. Also Anjali B. Okay, so you guys are correct. Tannins basically reduce the bioavailability of minerals primarily by chelating metal ions such as iron and zinc. So these polyphenol compounds basically forms strong and in complex, basically insoluble complexes with the minerals and preventing their absorption in the gastrointestinal tract. So this is particularly significant in the plant-based diets where tannins are evidently present, and it leads to deficiencies of the essential trace minerals. So their primary mechanism involves mineral chelation, leading to reduced mineral uptake. So option A is incorrect because tannins do not bind to lipids. Their primary interaction is with metal and proteins. And option C is incorrect because tannins do not directly interfere with protein synthesis. And option D is also not correct because tannins do not enhances oxidative stress in the gut but instead exhibit antioxidant properties. So here answer B is correct. So tannins basically are, there are many tannins. So they are classified into four groups. Hydro, I'm so sorry, it's very, I have to zoom it. So hydrosoluble tannins are there. So condensed tannins are also there, and then complex tannins and fluorotannins. So it is a classification of tannins. So there are four types of tannin, four groups of tannins are there. And if we talk about the bioavailability and bioaccessibility, so basically tannins, if we eat, so they are non-absorbable complexes, and there are some absorbable tannins are also there. So high molecular weight one are the non-absorbable, but the low molecular weights they are absorbable. So they can have some effects also because of their chemical structure or how they are formed. So here option B is correct. Tannins basically only work with the metal ions basically, and like the zinc and the iron mainly, it can reduce the bioavailability of these minerals. Okay, let's now talk about the pigments. So there are two types of pigments, natural and synthetic. In natural, we have organic and inorganic. So organic includes beta carotenes and lycopenes, and inorganic may include basically red earth pigment. and amber, like that. And synthetic pigments can also be organic and inorganic. So inorganics may include iron oxide, titanium oxides, like that, and organic may include indigoid or indigo, like other synthetic compounds, like that. And the natural pigments can also be classified based on their sources. So it can be sourced from plants or animals or microorganisms. So from the plants we get in carotenoids, chlorophylls, lycopene, and betalains. And from animals we get melanin, carmine pigment, sodium, copper, chlorophyllin, and astaxanthin. And from microorganisms, like different pigments we get, like phycocyanin, and we can also get prodiglosin and other compounds, like other pigments like monas monascus pigments, like that we can get. So basically based on the sources like plants or animals or the microorganisms, the natural pigments can be classified. So we have many natural pigments, and uh, they uh, they can be organic, also they can be inorganic, and we can also have synthetic one also. So let's discuss the first question about pigments. We have anthocyanin, betalain, carotenoids, and chlorophyll. In column 2, we have the pH dependent color properties, like stable across pH range of 4 to 6, olive brown under acidic conditions, red at low pH, blue at high pH, and exhibit minimal color change with pH. So we can see here from the picture that anthocyanin and betalains can be of different color range, then chlorophyll, and then anthocyanin, betalains, and carotenoids. So they can be based on different pH, they can be of different colors, and based on the chemical structure also, it can be of different colors. Shiva A, Anjali C. Okay, let's see. So C is the correct answer here. So we have different these pigments like anthocyanin. So basically its additive number E163, and it is red to yellow color, sorry, red to blue color, and it can be we can get it from black grapes, cherries, carrots, or strawberries. Betanins, it is red to pink color. It is get from beetroots, and its number is E162. And then we have chlorophylls, which is green in color, like alfalfa grass, nettles, spinach, and its number is E140/1. And carotenoids can be from yellow, orange, or red, like annatto, carrots, from tomatoes we can get, and it can be E160A to G. Anthocyanins, basically they are highly pH sensitive pigments, appearing red in acidic conditions and transitioning to blue or purple at higher pH levels due to the structural change in their basically flavium flavium cation. So basically there is this structure in anthocyanin, flavium cation. So based on the pH, it will change its color. So because of different present in different forms, because of present in different forms, it can have different names or different molecules, or and because of that there are different color from basically uh, blue or purple to red or pink. It can have these different compounds. These all are anthocyanin group compounds. So color changes in anthocyanin will result from molecular quinoidal or an hydrobases and then to the stable but slowly decomposing colorless carbonyl bases. So basically, because anthocyanin can have this flavium cation, so it can form different compounds, and based on that, there will be different colors of the compounds of anthocyanin. Betalins, they are found in beetroots or beets, and they're stable in the pH range from 4 to 6, but they can degrade at extreme pH levels, and it is divided into red, violet, beta cyanins, and yellow to orange will be beta xanthines. So this is basically betanin structure, which is basically found in the beets, and it is basically red glycosidic food dye. Is carotenoids. So basically, carotenoids are having different like structures, alpha carotene, beta carotene, lycopene, like all these, lutein, like that, beta cryptoxanthin. So these are different carotenoids. So they can be found in these different food products like papaya, tomato, watermelon, carrots, pumpkin, like those we can get like oranges. So carotenoids such as beta carotene, lycopene, they exhibit minimal color change with pH variation as they are lipid pigments and do not interact significantly with acidic or alkaline environments. Carotenoids basically are terpenes and they exhibit many colors orange, red and purple. So this is the structure of beta carotene, this is the structure of alpha carotene. So basically, next is chlorophyll compounds. So basically chlorophyll, they are basically porphyrin compounds which are having macrocyclic tetrapyrrole pigments in which the pyrrole rings are there and joined by the methine bridges. So basically, and it is having the basically double bond system forms a closed conjugated loop. So this in a centrally located basically magnesium iron is basically coordinated with the four pyrrole rings of the nitrogen. So we have already seen the structure of chlorophyll before. I hope. So you please find the structure of chlorophyll. I don't know where we have discussed the structure of chlorophyll. So basically, in chlorophyll structure, what happens is magnesium ion is there, a magnesium atom is there, and there will be four nitrogen atom will be there, and these will be attached to the pyrrole ring. Okay, so we discussed basically the vitamin B12 structure, which is cobalamin, in which in between the cobalt is there. Here, magnesium atom is there, and which is attached to four nitrogen groups, and which is forming the coordination bond with the four pyrrole rings. Like this is the chlorophyll structure. So the chlorophyll basically is turned from green to olive brown in the acidic condition. So if there is acidic condition, what will going to happen? The magnesium ion which is there in the at the middle will going to remove. So it will become basically the pheophytin kind of molecule it will form, and because the magnesium ion is at the center is basically replaced by hydrogen, the green color will be converted into olive brown color. So basically chlorophyll has two compounds, chlorophyll A and chlorophyll B. Chlorophyll A is blue, this is chlorophyll A which is blue green, and chlorophyll B is yellow green. So we can see in the visible spectra 400 to 700 nanometer, these are there, and Carotenoids are basically yellow color. It shows this peak. So here in this question, anthocyanin is basically associated with the red at low pH and blue at higher pH. Betalains, they are associated with stable across pH range 4 to 6. Then carotenoids, they are exhibit minimal color change with pH, and chlorophyll are associated to olive brown under acidic condition because it form basically pheophytin group, which is this, and where this magnesium ion is not there, atom is not there, and it is replaced by H. Next question is chlorophyll A differs from chlorophyll B by having which group instead of a formyl group at position 3? Hydroxyl, methyl, vinyl, or ethyl. So basically chlorophylls are forefronts, and it is having basically this structure Mg at the middle, at the center, and with N there are 4N which is coordinately bonded. With that, pyrrole rings will be attached. 4 pyrrole rings will be attached. So how chlorophyll A structure is different from chlorophyll B structure? Anyone who knows the structure of chlorophyll A and chlorophyll B? Anjali is saying C, Shiva is saying B. Okay, Anjali is saying C in the previous question. Okay, Shiva B, Kishore group basically. So you both guys are correct. Methyl group is there. So in chlorophyll A structure, if you see, there is this methyl group, and in chlorophyll B, we have the formyl group at carbon position 3. So we have chlorophyll A and B, which are the two types of chlorophyll found in plants. So it differs in their side chain at position 3 of the chlorine ring. So chlorophyll A has methyl group CH3, whereas chlorophyll B has formyl group CHO, which is this group. At this carbon, these two groups are different here. This structural difference variation in their light absorption property. So chlorophyll A basically absorbs more light in red region, whereas chlorophyll B in blue region of the spectrum. The hydroxyl, vinyl, and ethyl, but they are basically not at position 3 in chlorophyll A and B. So answer B is correct, methyl group. Which of the following pigment is responsible for the bright red color of the fresh meat? Met, myoglobin, oxymyoglobin, nitrosyl myoglobin, and sulfomyoglobin. So which is responsible for bright red color of fresh meat? Anyone who is answering this question? Anyone who would like to answer this question? Shiva B, Anjali A, Anju A. So we have A and B as our two guesses. Let's see. So oxymyoglobin is the correct answer. So oxymyoglobin is basically the right is responsible for bright red color of the meat, fresh meat. it forms when myoglobin, so basically myoglobin molecule, which is a protein, it will bind to oxygen in the muscle tissues and interact with the oxygen to create a stable oxygenated compound. So oxygenated compound will form more stable, and this bright red color is often associated with the meat freshness and is desirable for the consumer. So basically myoglobin is getting attached with the oxygen and forming a oxidative compound which is known as oxymyoglobin, which is responsible for the bright color of the meat. So if we see here, basically the myoglobin, which is in purplish red color, so meat will be in purplish red color, it will be getting oxygenation and it will convert into oxymyoglobin, bright red color we can see. So after that, metmyoglobin is formed. How? When myoglobin is oxidized and it will convert into brown color during the spoilage or aging process. So what will happen? This oxymyoglobin is further oxidized into metmyoglobin and it will form brown color, which is the significance of that it is the meat is getting spoiled or it is getting uh, aged, or so it's not desirable by consumers. Next is the nitrosyl myoglobin, which is a pink colored pigment and it is found in the cured meat due to the reaction of myoglobin with nitrates. So myoglobin, if react with nitrates, it forms pink colored cured meat, which is because of the formation of the compound of nitrosyl myoglobin. Basically, if we are adding oxygen at sixth position of the heme, that is oxygenation, so we are converting into oxymyoglobin. And deoxygenation matlab if we are removing the oxygen. So basically these are deoxygenating. So next is the sulfomyoglobin. So sulfomyoglobin is basically when we are getting the red pigment, basically sorry, green pigment basically. So meat will be having green color of pigment or green color will be there because what will going to happen, this myoglobin, when it will attach with the sulfur containing compounds or interact with sulfur containing compounds, the meat will going to be spoiled, and because of that green color we can see, and that is because of the production of sulfomyoglobin compound. So basically myoglobin is here. So when it is interacting with nitrate compound, it will form nitrosyl myoglobin. So it will have the at time pink color like that will get. But if we are adding with the hydrogen sulfide or sulfide group we are adding, so green color compound will be there, which is the spoiled milk is. So spoiled meat is there. So here B answer is correct. Okay, let's talk about dyes now. So dyes can be two types, natural dye and synthetic dye. So natural dye can be from vegetable, from insects, from minerals. So basically this dye vegetable we can get from stem, from roots, from flowers, from leaves, and dyes we can get from insects like lac or other dyes like tyrant dye, purple, like that. And from insects if it and from minerals if we talk about, it's gold buff, iron oxides, so from there. And synthetic dyes may include water soluble and water insoluble. So water soluble are the can be direct dyes, acid dyes, basic dyes, or reactive dyes, further classified into these dyes. And insoluble, water insoluble can be classified as wet dyes, dispersed dyes, and sulfur dyes. So these are the different dyes based on their origin. Now we are discussing dyes. So let's discuss this question. Which among the following is resistant to acidic conditions and high temperature? So we have this, then corn, sunset yellow, and brilliant blue. So which is resistant to acidic conditions? Anyone who would like to answer? So here we can see from the picture that this is the food which needs dye or the pigments of the colors. So like these candies are there in which we add these pigments. So this is basically first is the tetrazine, which is of the yellow color, and then oncure, which is of red color, and then sunset yellow of slightish orangish color, and the brilliant blue is of like this color. Shiva, sunset yellow FCF. Anyone else? Resistant to acidic condition and high temperature. Okay, let's discuss the solution part. So tetrazine is the correct answer. Tetrazine, which its E number is 102, and it is a synthetic and water-soluble monoazo dye. So it is widely used in acidic and heat-processed foods, basically due to excellent stability under such conditions. It retains yellow to orange color in low pH environments, making it a suitable for beverages and baked goods. While this second one, Ponche, this is strawberry red azo dye and sunset yellow, which is orange azo dye, are basically synthetic dyes but less stable in highly acidic conditions and high temperature conditions. Ponque basically is going to be fed when there is ascorbic acid. Whereas brilliant blue dye, basically which is blue color and water soluble, it is basically used for blue shades and does not have the good stability under the acidic conditions, such like tetrazine. So it remains basically tetrazine, which we, when we have the acidic condition, we use this dye. So next is the classification of the flavor compounds. So our last topic for today is flavor compounds. So basically based on the chemical structure, so the flavors are divided based on the structure, like if they have the alcoholic structures or they have ester or pyrazines or ketones or aldehydes or fatty acids or aromatic compounds or lactones. So they are classified into from these classifying. So they have different like many like high very good number of the flavor compounds. So we are just discussing that based on these are the classification basis. We are not talking about each one in detail right now. So we have the flavor compounds in column 1, citral, vanillin, eugenol, and benzaldehyde. In column 2, we have the sources like cloves, cherries, lemons, and vanilla. So we need to, so this is the vanilla. This is lemon, this is cherries, and this is cloves. So we need to match the flavor compounds with their sources. So this is a very simple question. Maximum of us are saying B. Okay. So B is the correct answer. You all are correct. So citral is basically a terpene which is having a strong lemon scent and is found in the lemons. Vanillin is basically responsible for the characteristic aroma of vanilla, and it is derived from vanilla beans. And eugenol is basically with the spicy and warm notes, so it is derived from cloves or the spices. And benzaldehyde is like almond-like aroma, which we are associated with cherries. So artificial cherry flavoring um, is the benzaldehyde. So in the phenol classes, basically phenols are the compounds that contains a hydroxyl group bonded directly with the and aromatic benzene ring. So in this, if we talk about here, the eugenol, which is the active compound of the clove oil, is there. So eugenol imparts warm, spicy aroma, key clove oils. So if we talk about the citral, so citral is a aldehyde group of compounds. So aldehydes are also organic compounds, but they'll have this carbonyl group which is bonded to at least one hydrogen atom. So citrals, they are basically key flavor components in lemon, and citral basically consists of the two isomers ger, which is gernaline and narale, and it is also contributing to strong lemon taste. Another is a cinnamaldehyde class, which is in the aldehydes only. So it is basically uh, in the cinnamon oil we can find, or usually it is having um, strong aroma. And benzaldehyde basically it mimics the cherry flavor and widely used to produce cherry flavor. Uh, even it is not naturally available in cherry like this. This is the and vanillin basically is basically a compound which is basically having the vanilla kind of strong scent, strong flavor. And if we talk about other compounds which are found in other organic food products, so it is like in apple there is hexanol, cream, cucumbers, chocolate. So they'll have different aldehyde kind of compounds which are there to which is producing the flavor. So here citral is related to lemons, vanillin is related to vanilla, eugenol is related to cloves, and benzaldehyde is related to cherries. The flavor of blue cheese is basically due to the presence of eugenol, citral, 2,3-butanol, and 2-heptanol. Which one is the correct answer? Okay, so let's see the answer. Shiva C, Kishore C, and Sainath also C. Okay, 2-heptanol. So this is the blue cheese. So basically, you guys are correct. Heptanol is the correct answer. So it is basically a ketone kind of organic compounds. So it is bonded. This carbonyl group is carbon atoms where this 2,3 butanen dione is basically this 2,3-butane-dione is basically responsible for the buttery flavor, uh, or celery, or for many other compounds, flavor of butter, celery, and many other foods. So this two heptanone and two nonanone, basically they are the flavor compounds of the blue cheese. So this is the basically for the blue cheese. This is so the distinct flavor of blue cheese are basically because of the two heptanone, a volatile ketone, which is produced during the metabolic activity of the molds, like penicillium roquefort. So basically, when cheese is ripening by this particular strain of the penicillium, so what we're going to, because of the metabolic activity of this molds, this compound releases. So imparts strong, tangy, and slight pungent aroma, which is characteristics of the blue cheese. Whereas 2,3-butane-dione is basically contributing to the buttery flavor in other dairy products and is not significant contributor to blue cheese flavor. Eugenol, we discussed about that it is providing the clove-like aroma, or in the spicy food it is associated, and citral is associated to the citrus scents, like it is found in the lemons and the lime oils. So today we discussed about vitamins, like importance and roles of vitamins in human body and human nutrition. And we discussed some of the structures and the functions and the deficiencies of the various fat solubles and water soluble vitamins. Then we discussed about vitamin, like when we are processing vitamin C, stability when we are using the heat or other processing conditions cooking. Then we discussed about minerals, about macro and micro minerals, and their help, how they are going to control the metabolism, or also when we have this minerals uptake, so how they will help in the body regular function, and if there is deficiency of these nutrients or minerals, micronutrients, so what is going to happen. Then we discussed about phytochemicals and bioactive compounds and their bioavailability and their absorption. So we discussed about isoflavones, we discussed about tannins and other compounds which are responsible also hampering the bioavailability of other minerals and all. So we discussed about pigments and the colors, like the natural and the both the synthetic one. So we discussed about the synthetic brilliant blue, like those, and in natural we discussed about anthocyanin, myoglobin, and we discussed about the chlorophyll compounds, we discussed about carotenoids, lycopene, and we discussed about flavoring compounds, both natural and and synthetic and natural identical. So we mainly targeted the discussion around aldehyde ketone group of the compounds. So in the next class, we will be discussing about food microorganisms, microbial food, microbial growth, and how they are spoiling the food products. And then we will be discussing about how to prevent the food poisoning and spoilage, and what are the different beneficial microorganisms which are there needed in the food. So I'll be uploading the respective study materials on the sites. And if you have any question, feel free to ask. Thank you so much for joining today's class. Let's meet on next Sunday on the same time, 4 to 6. So if you have any questions or any doubts, you please stay. Otherwise, you can leave the meeting. Shiva, thank you so much for joining the class. Sainath, thank you so much for your feedback and thank you so much for joining the class today. Kishore, thank you so much for joining the class. Hope to see you guys in the next class. If you guys have any doubt, please ask your questions. Otherwise, you can leave the meeting. Anyone is having any doubt? So I'm stopping the recording now.